Today I'm going to talk about the microbiome. This has been a topic that I've wanted to speak about for too long now. When I was first trying to communicate this topic, I was trying to use the analogy of Star Wars, and I'm still going to use that today. When Qui-Gon first announced the midi-chlorians in the Phantom Menace, and this isn't going to be a video about Star Wars, but it's just where I'm starting this off with, okay? And Qui-Gon specifically says, life forms living together for mutual benefit. That was symbiont. So the analogy is how we are living in a symbiotic relationship with them. The reason why I think that that's a really useful analogy is that it's very similar, in my understanding, as the microbiome. So the vast microbes that are inside of us. More particularly, I'm going to speak about bacteria in this video. Essentially how we communicate with these life forms. They continually speak to us, showing us the will of the force, if you know. And that is the topic I'm going to dive into today. I believe I had this idea in my head because I was reading Dr. James Kinross' Dark Matter, and this was where the analogies start to come into play, because I think I rewatched Star Wars at the same time, and I was like, well, mind blown, like these references are like the exact same. He likes to use metaphors that are very spacey. He says human galaxies and interconnected organs. The organisms inside of us, the organisms produce dark energy, a chemical and immunological force that sustains us. And this is the main important bit that stood out to me. These organisms will ultimately destroy us if we allow them to become unregulated to the dark side. So fast forward to October, and I stumbled across this 2025 paper. It's a very impressive one. It was also a very important part of a very important editorial. And it was within Women in Infectious Agents and Disease 2024. I will link it. So they found that even commensal, commensal means our good bacteria. So there's 3 different terms for how the bacteria's trait. And that's commensal, opportunistic, and pathogenic. Commensal is the good bacteria. Opportunistic, I nearly went like that, didn't I? Opportunistic is a commensal bacteria which could have the potential to become pathogenic, so your bad bacteria, and then you have strictly pathogenic bacteria, which are just like, lo and behold, bad. And what they found is that good commensal intestinal bacteria, E. coli, carries pathogenicity potential, and then it's almost impossible to tell the beneficial from harmful gut bacteria apart. And this was, as such, my research frenzy into the complexity of the microbiome in health and disease. And this is what it took me to up until now. I began to question, because of this study, how there was like little differentiation between good and bad bacteria, how a language communication between our bacteria and us, if this is similar to the gut-brain axis, where I had studied before how your neuroendocrine system can affect your gut microbiome, and your gut microbiome can affect your brain. It's the gut-brain axis, right? So I thought, if there was no distinction necessarily between in commensal bacteria being good or bad, how might we influence that process, and how might that process influence us? If you see, how does it fall into that ball game, basically? So in Dr. James Kinross's book Dark Matter, it was the first time that I had heard about quorum sensing, or at least, forgive me, all of my lecturers at university in microbiology, if you had brought this up before, because I swear it's only become fully interesting to me now. But maybe that was like a key code. It's a form of molecular communication among bacteria that becomes activated when a population of the bacteria reaches a certain density, and he described it as a language that they use to communicate with their hosts. So once there's enough of the bacteria and there's a population, then they start releasing these things called autoinducers. to be specific, it's a cell-to-cell communication process in which bacteria detect and respond to hormone-like signaling molecules known as autoinducers. But this system can also participate in inter-kingdom signaling, so that is between the host and bacteria. I struggled to get my head around it as much as like the next person. It's on par with like quantum physics, I swear. It does take a while to get used to, so I'll try and explain it as best as I can. Okay, so very simply, quorum sensing is how bacteria sense what's going on around them and how they decide to behave. They do this using sensors and receptors, and one of the receptors is called QSEC, and it's interesting because it can listen to two different kinds of signals. So on one side, it listens to signals from other bacteria, and these signals build up when bacterial population density increases, and that is classic quorum sensing. Then on the other side, the same receptor can also respond to signals from the host, so specifically stress hormones, adrenaline and noradrenaline. This is called inter-kingdom signaling. And different signals can be interpreted by the same receptor, so this QSEC. When either of those signals are detected, QSEC triggers an internal response that can switch on genes associated with virulence in certain strains of bacteria. So rather than bacteria behaving in a fixed way, their behaviour depends on the context, so population, environment, and host signals. So you've got two pathways with the same outcome. You've got quorum sensing, autoinducer signals, and high bacterial population associated with the bacteria. And then we've got the stress hormones and the interkingdom signaling. And this is coming from the host. And both of these things can lead to, let's say, gene activation and virulence-related behaviour. So there's also research to show that both commensal bacteria and pathogenic strains can synthesize the same signaling molecules, including AI2 and AI3. So the AI2 was related to the population density, and then we've got AI3 that was related to the inter-kingdom signaling. So it's another study that supports this. And then when we move on to the 2025 study, it's just to showcase that it found how commensal E. coli from healthy adults also carry traits that we usually associate with pathogenic strains. So that would be these virulence genes. While that doesn't mean these bacteria are causing disease, it does mean that there is a capacity for certain behaviours to exist across strains, and whether those behaviours emerge depends on the context. So, it's seen that there were two factors that could tip the bacterium into activating the virulence genes. That being a disproportion of bacterial population, and this can also be considered dysbiosis, right? So it's when you have too much of a population over the other. So there's a disbalance, there's dysregulation, there's a accumulation of one particular bacterium over another, alongside the stress hormones as well. And together, boom, virulence factors. Going back to the 2025 study that showed how these virulence factors are present in the commensal, the good bacteria, right? This means it's not as simple as eradicating the E. coli from our bodies. The potential to become pathogenic exists out of the state of infection. This is just what happens in our normal everyday life. Like, we have the potential to have these virulence factors because it's the good bacteria that already exists within us and is actually beneficial to us. And going back to Dr. James Kinross, he specifically said as well, bacteria aren't inherently good or bad. Their effects depend on the species location, balance with other microbes, and the health of the host. The studies that have been done regarding virulence factors and quorum sensing are what's called in vitro, which is where it's done in like a little petri dish and it exists outside of the body. However, bacterial virulence, as we've just disclosed, is context dependent. It's not fixed, and once again, the microbiome is considered a complex and dynamic system, similar to when I was talking about chaos theory and neurological science in my previous two videos. I believe this is where my theme is going now. I'm just going to advocate complexity science forever. And as such, because of the system and the way it behaves, it would resist any sort of deterministic or reductionist approach. That being said, in the 2025 paper as well, their concluding remarks were, the host environment, including immune responses, nutrient availability, and physical interactions, often influences gene expression in ways that cannot be entirely replicated in vitro. In vitro studies' first step before advanced research can provide valuable information on expression of virulence factors. So I cannot wait, and I long for the day that I can get involved in these type of experiments, because this is the future for me. So what we do know is there is a molecular language. Bacteria can communicate with each other and with us as the host. We are symbiotic beings. They can communicate with each other and they can communicate with us, responding to our signals and to the environment. This part isn't speculated. What seems clear is that in a symbiotic system, health and disease are not fixed states. They are context-dependent behaviours. Could raise questions about lifestyle factors such as stress and diet, but I don't think I'm gonna use this video to answer those just yet, because the information is just not there yet. But just maybe, this study could highlight the importance of acknowledging a holistic view in research and can consider how communication within and between species might lead to unexpected or paradoxical effects. Until next time, I have another Star Wars analogy coming up. Maybe one more after that too. Let's try this again, shall we? Approaching this study, this entire analogy. But maybe this book, this study of acknowledging. Oh wow. All right. AI2 and AI3 that I've just mentioned.